important. Depending on what you're doing, you may care more about one instrument or another, or what kind of music. The same kind of thing takes place in ENCODE. So the reason for ENCODE, I think, is quite clear. The completion of the Human Genome Project became obvious that understanding parts of the genome is difficult. Understanding what portions of the genome are doing something of value is difficult. We have a genetic code that tells us about protein coding regions. It's a very small part of the genome that's very sparse, right? So how do we find the regulatory regions? How do we find out what they're doing? And how to identify them is another problem. And this is important because we know that non-coding DNA does a lot of important things. The vast majority of common disease associations and heritability of the associations is imputed to lie in non-coding regions, so that's important. There are Mendelian disorders that are caused by genetic variation in non-coding regions. So by those two measures, among others, non-coding DNA is important. But it's not easy to study, and one of the dysfunctional information could be used to interpret disease, gene regulation, mechanisms of mutation, and so forth, if we had a better understanding of it. So ENCODE is one project. A lot of individual investigators are also working in this area, trying to get a better understanding of these issues. Here's a graphic way to see the problem. You just take a snippet of the genome, not a random snippet, but snippet. Looking at it quickly, it's difficult for most people to get information out of it. Now, to some extent, this is an exaggeration, right? We all know that partially this is a solved problem. You can have apps like at the bottom of the slide where you can mark exons and you can mark variants that are associated with disease, or you can do the same thing by color coding the sequence. And that gives you some information now that you didn't get just from looking at the sequence. The approach that ENCODE is taking, a lot of individual investigators doing this as well, is to build richer maps so that we might have more information. So how this might work is now you can see the same GWAS variant in context of the gene. This comes from non-ENCODE work. And from ENCODE data, you can see that there are biochemical signatures in this region. We'll explain what they might look like. And it turns out that this is a signature for a candidate regulatory region. So that tells you something about this variant could lie in a regulatory region. ENCODE data can tell you other stuff because it's cell type specific. You can see that this information is preferentially coming from a particular cell type. So that may give you some idea of what cell type is important in this process. And while this variant appears to lie within a particular gene, you can see that easily from sequence alignment. There are ways to predict what gene it might be working with. This is, of course, predictive, not with certainty. And in this case, the prediction is that this is a regulatory variant is actually working to control the neighboring genes. So being able to have these stories moving forward would help us to much better appreciate the role of genetic variants and phenotypic traits. So the goals of ENCODE are really simple to state how to get to this story. We're trying to identify all of the candidate functional elements of the genome. We're also trying to share this resource freely with the community. Freely means it's unrestricted access, not controlled access. There's no login, there's no purchase. You simply go to the website, use whatever it is that you want. And people are using this to look at the genetic basis of disease, gene regulation, how mutation occurs, and so forth. But any process that you'd like to use it for is allowed. So the way ENCODE is doing this resource is built upon decades of research in gene regulation. For years, people have known different biochemical signatures are associated with gene regulation, transcription, post-transcriptional processes. And these have been converted into genome-wide assets. And then the results of these are used to sort of reverse engineer to make predictions about how the genome is working. So you can make predictions about where genes are, it's probably the simplest part, what are the transcripts that are encoded within those genes, and then what are regulatory elements such as promoters, enhancers, and other distal regulatory elements that might be controlling those genes. So a big shout out to the gene regulation community that has over many years to develop these techniques. And ENCODE data are very cell type specific, which is both an advantage and a drawback to doing this. It's a drawback because you need to collect the data in many different cell states, unlike first approximation with DNA sequencing. But then it gives you richer information because the answers that you get back tell you something about what cell types are involved. And if you look at differentiation over time, total cells start off with one set of candidate regulatory elements. Some of those are extinguished over time. New elements appear to be turned on in progenitor cells, and then other elements turn on in fully differentiated cells. So these changes in regulatory element signatures tell us about cell type. So to date, ENCODE has done a lot of useful things that has helped out the community. We're sharing thousands of data sets. Today they're shared without any embargo. There's no wait 1 year until the consortium has had a chance to publish on it today. They're shared through unrestricted access, the equivalent of GEO. There's no signing up for DataGap or buying an account or something to access the data. The data are uniformly processed to a large extent, which minimizes the chance of coming up with cross-data set comparison and finding differential effects that aren't real or analysis artifacts. We're also sharing the software that we've either produced or used in the project so others can use it and also make things transparent. And we're working with other projects, other individual investigators to make the data more interoperability, talking about standards and how things are communicated. One of the things that we share are ENCODE publications, and there are hundreds that have come from the consortium. But perhaps for this audience more important, there are over 1,000 publications from outside of ENCODE that have used ENCODE data. Some of these look at human disease, some of these look at basic biology, and they cover a wide spectrum of diseases. And I think that this attests to the translational value of the resource. So in summary, we're trying to create a catalog of all functional elements of the genome. This is freely shared with the community. The foundation for this is established techniques and methodologies that have been used for years in gene regulation studies. And these maps can be used to make predictions about genome function. This should help you know the work. So now we're going to take a quick peek at how you might use ENCODE to study the role of genetic variation in human disease. And here I'm going to give you a few use cases, some ideas that I've gleaned from the literature. This is not the only some whole of what can be done. The major idea here is that ENCODE supports hypothesis generation and refinement. Okay, high throughput data are useful in identifying hypotheses tests, and some of these things are predicting causal variants for traits or diseases, predicting target genes for either regulatory elements or genetic variants, cell types that are affected by regulatory elements or variants, and then also predicting upstream regulators, understanding pathways to how these systems work. The prediction of causal variants, some of it's obvious problems, others it's not obvious that this is an issue, but everyone does a study such as GWAS, you get a locus, but there are other variants that are in LD, so that doesn't tell you, just from that first step, what is the causal variant. ENCODE type annotations can be used as one line mapping strategy. You've also got the issue that it's possible that multiple variants work on the same trait or disorder. You might not be trying to find the causal variant, rather a group of them. So ENCODE data can help you to figure that out. And a recently published study looked at ENCODE and Common Fund roadmap epigenomics data, found that many GWAS findings overlap with these annotations. So that suggests that this could be used. If one is not familiar with these kinds of assays, the simplest way to get started is there are a couple of pre-computed resources. First, I'll tell you about HaploRank from the Noah Ellis group at MIT that can help you to sort through ENCODE and roadmap data. And with HaploRank, you can, step one, put in genomic coordinates or SNP IDs or list of them and then click submit. And then it will return pre-computed analysis of ENCODE data. This is just the first step of the kind of result that you get returned. And it'll show you for both the lead SNP and others that are in LD what kinds of ENCODE and roadmap annotations are found in that area. And that can help guide you in further experiments to decide what you want to do next. Similarly, Mike Snyder's group and Mike Cherry's group, both at Stanford, developed Regulome TV, and in a different way, this analyzes ENCODE data, roadmap data, and some other data sources. Again, you can put in genomic coordinates or SNP IDs, and then click Move On, and initially you're presented with a score, the likelihood that the genomic region you're looking at has regulatory potential, and then of course you can drill down and see the individual underlying data and see why that assessment came about. As Gene said at the beginning, and I meant to say at the beginning but forgot, please don't worry about writing down URLs or detailed explanations. All of these slides are going to be shared, so that information will be available to everybody. There's also ENCODE Syselement browser, and this has a number of different functions for human data and mouse data. And in this example, you can again put in a genomic coordinate, and it will find candidate regulatory elements. In this case, it finds them by looking for DNA hypersensitive sites, and also tell you about candidate upstream regulators by looking for motifs that are found in those regions. And prediction of target genes is also an interesting problem. GTEx and ENCODE data and other projects are finding that it's not unusual that regulatory sites don't work on the nearest gene. They work on some gene that's further away. So one finds a piece of DNA that you think is important, and it's not protein coding. If you just guess that the nearest gene is its target, the growing story is you'll be wrong at least half the time. Okay. And if we look at, for instance, predictions based on GWAS studies, a lot of times it's quite just that the regulatory region is from the gene. And in a practical sense, what this could mean is you might have some variant of interest and start studying the nearest gene. But perhaps there's a better candidate that one could work with, a better prediction, and that could change your research project if you didn't have to spend a few months studying one gene, studying the next group, or only filter was proximity. If you knew other principal ways to say what was the likely candidate, you could come up with a better answer. So you can do this by using regulatory elements database. Terry Furey's labs developed this, and it works a number of different ways. One of the things you can do is put in genomic coordinates, and it'll again report DNA hypersensitive sites in that region, candidate regulatory regions. And if you click on any of them, it tells you additional information. And one of the things it will tell you is if there's a prediction for what gene is the likely target of that, will give you a confidence estimate and what that target gene might be. The ENCODE Syselement browser, which I pointed out before, and in the bottom of these slides you can see that you're going to be hearing more about this in workshop session 3, for example. So this is just a very quick overview, but again, you can put in a gene name, and this tool will return to you what are regulatory elements that appear, candidate regulatory elements that might be linked to this gene. Okay. And the one that I've highlighted here is one that I showed in that initial example, showing a common control region that affected many genes. I chose that region, that example, because it knows that it's been very well worked out, that region's been knocked out, and it does in fact control these genes. If you go to ENCODE human data, you could make the same prediction without having done any of them. And the last use case I'll tell you about is predicting target cell types. Again, to some people this is surprising that this is something you would need to do, to others it's intuitive. But if you think about most human diseases affect several cell types. Sometimes they affect several cell types directly, sometimes it's more subtle. For instance, sometimes the affected cell type is a developmental niche for the cell type that actually shows the dramatic effect. There are people who have lack of lymphocytes because their thymus, the niche where lymphocytes develop, is impaired. Right? So the affected cell type is not where that gene appears. And sometimes, of course, the etiology of diseases is not well understood. I'm old enough to be able to remember when it was a wild and crazy idea that type 1 diabetes, a metabolic disorder, might involve the immune system. And we now know it's an autoimmune disorder, but for some fraction of diseases that we work on today, we don't know the cell types. So again, in regulatory element database, if you call up different elements, in some cases, those elements appear to be active in only some cell types. Other elements appear to be broadly utilized. So that could give you some information about cell type that's affected. Similarly, if you look at haploarray and regulon DD, which I talked about briefly, when you see the underlying evidence, if for instance you mouse over something in haploarray, it tells you the cell types that that data came from. And that could guide your hypothesis, especially if something's known about the disease. You think Alzheimer's, okay, neurons, macrophages, glial cells, and your hit is for glial cells, right? Then that helps you refine things. It's more difficult to use if nothing is known about the disease. And if we look at the ENCODE syselement browser, and for instance look at gene expression, you can type in a mouse or human gene in. It might be the gene that you think is important in that disease. This is expressed in restricted number of cell types. Obviously some other genes are broadly expressed. And that again could guide your hypothesis as to what's going on. A more principled way to do this is you could take you could intersect genetic variants that are linked to the disorder, so intersect them with regulatory elements, and then ask whether those regulatory elements are enriched in particular cell types. And the code for doing this is shared in this URL on the slide, and then you can ask in a more principled way which of several candidate cell types might be involved in a particular disorder or process. So in summary, I think the main thing that ENCODE can help with is hypothesis generation and refinement, and that this can be used to predict causal variants, to predict target genes, to predict cell types and upstream regulators. All of the examples I showed you are examples of how you could use this with genetic results. If you had epigenetic results or an epigenetic cohort, you could do many of the same things with ENCODE data. Of course, if you're skilled in that, then you know there's some additional caveats apply. The same kind of thing I showed in germline examples, but one could do all of this with somatic variants, but again, additional caveats apply when you have that. So now I'm going to quickly run through how you can access ENCODE materials. You're going to hear a lot more about this, at least in workshops 2, 1, and 5. So the ENCODE portal, encodeproject.org, that's the one URL I would want you to remember, encodeproject.org. And it has a lot of this info. And for instance, you can access the data under either search or matrix. If you go to matrix, there's a nice representation, which I've crudely trimmed so you can't see most of it. It shows you the kind of data that's offered, data types, projects where the data come from, and so forth. And you can use facets to narrow down the subset of the data that you want to see. I want to emphasize the data policy. As I said in the beginning, this is freely shared. So if the data are there, you can use it. There's no restriction, no embargo or anything. If you ask a standard scientific thing, that cite the project, if you, like with any other data source, you can use something, cite it. For reasons of transparency and reproducibility, it's good to share accession numbers. If you share the accession numbers of what you did, then your peers can tell what you did and how you did it. Okay. New to the ENCODE portal is the search by region. You can put in a genome coordinator SNP-ID, find ENCODE data that overlaps with that region, and visualize it. That's a quick way for those of you that want to see trends or to download elements to get your data. There's an ENCODE encyclopedia. You're going to be hearing about this in the focus of workshop 1. All right, and it has high level and very close to the data analysis, has a range of things. Ground level, close to the data annotations are things like where the transcription factors bounds. Middle level annotations are putting the data together to find things like where are promoters and enhancers and how can you visualize them. And then top level annotations put together many different types of data to generate inferences. You'll be hearing about that, especially in workshop 5. ENCODE publications. Again, there are community publications that I would point out to you because they're examples of how people outside of ENCODE are using the data to study human disease, to study basic biology, to produce software tools. There are also ENCODE print publications that again illustrate what was done and how the data can be used. We're very interested in sharing standards for how things are done, and this is done again for the same 2 reasons. For transparency, so that you know what was done and how it was done, so you can evaluate for yourself how credible this is. And then we're also sharing these standards because there are many people in the field that are, including some that are newer in the field, and they're wondering, how should I be doing this? We hold this out as a way that you can do it, but of course it's for each group to decide how to do it. We don't dictate standards to anybody. So you can click through and see different types of standards and experiment documents that we share. We're sharing software that we're using in the project, again, so that people can understand what was done and how it was done, or if people are doing this kind of work and want to use software. Some of this software was created by ENCODE, some of the software is created outside the project, but we use it heavily, so we're both important to know about, and we share both of those kinds of software. And lastly, I'd like to give a shout out to our partners with the International Human Epigenome Consortium. ENCODE is a member of this. This is the NIH Common Fund Project Roadmap Epigenomics. A bunch of like-minded projects are collecting epigenomic data across many cell types and sharing them through different mechanisms. The ITAC data portal and project summary are available. There's lots of useful data there, including a subset of the ENCODE data. So I want to finalize here with a list of URLs. I don't expect you to read now or write down there. Again, all of these slides are shared. Click on the links rather than write them down. But we share, for instance, tutorials from the project, including the results of this will get posted online, analysis tools from the portal, and then I have resources. I highlight the ENCODE mailing list. If you want to hear regular updates on the project, you can join this list. So I'll conclude by reminding you that the goals of ENCODE are to find all of the candidate functional regions in the genome. This is an aspirational final goal. I think there's no way to know when it's done or how it's done. And we're freely sharing the results that we find as a resource for the community. And ENCODE data are already being used in studies in disease and human biology. And it's used any way that anyone can come up with. I'll stop here by thanking the people on the ENCODE consortium. There's another representation of some of them. I have the privilege of working with smart people every day and hearing great ideas. And it makes my job really fun. And my program colleagues at NHGRI, Elise and Peter, who have started the ENCODE project long before I had anything to do with it. Dan Gilchrist and I work on that project now along with Elise. And Dan's here today. I urge you to say hi to Dan as well. And they're great colleagues and people to work with. And of course, all of this work, like any other bit of science, is built on years of study that came before it. Lots of work on gene regulation, lots of work on genetic basis of human disease. So I'll stop there and take any questions if anybody has any questions, or we can move on to the next one. So now what I'd like to do is introduce our first scientific speaker, our keynote speaker. Oh, I have one question. Thank you.
So regarding the ENCODE feature, so what NHGRI plans to do? Like, what kind of focus or emphasize?
Sure. Some of this is public record, and some of this is to be decided yet. And I can tell you about the public record part. And we've recently accepted applications for a new round of ENCODE. They haven't been reviewed, the decisions haven't been made. But the applications we accepted were for new mapping centers, sort of like the production centers that we have now. Computational analysis centers as an ongoing activity we have now in ENCODE. And then a new type of activity, functional characterization centers. We also have applications for those, in addition to a data coordinating center and a data analysis center. So there's a good chance that moving forward, there'll be more data production in ENCODE, more mapping. There'll be releases of data through the DCC analysis. And then we might have additional information about function in different genomic regions. Past iterations of ENCODE did not focus on functional analysis of the mapping results. The thought was that was within the purview of the community. The idea was to do the mapping and to come up with raw material for others to work on. But we've seen that this is a need for us as well to be involved in, so that's what that's coming. To our keynote speaker, we're honored to have Nancy Thomas here from Vanderbilt, and she's going to tell us about her recent work looking at genetic contributions to gene regulation and how learning about that can help us understand about the role of genetics in human traits and human disorders. So please join me in welcoming Nancy. Thank you.
Thanks. It's a real pleasure to be here. This is such a great meeting, and I've been really looking forward to sharing some of the newest findings. I really want to get some feedback from an audience like this one and more collaborations with my ENCODE colleagues, because while this project was very much grounded in and started by research that we were doing in GTEC, it is very much going to end up in deeply looking at ENCODE. So what I'm going to do today, so I think over about a year ago, I was talking about initial results in about 5,000 subjects from BioView. And what I'm going to talk about today are results of studies that we've been doing with our PredictScan methodology in about 20,000 BioView subjects on our way to about 120,000. So there'll be some novel genes and phenotype discoveries, and more on the continuum from Mendelian to common disease in a number of different dimensions. Some new ways that I think we have to really get more kinds of insights into biological mechanisms of disease, and new big picture biology that relates to the medical phenome as a concept in and of itself, separate from individual diseases. So just to review, a lot of what I'm going to talk about is centered on this analysis tool that we had worked on through GTEC called PredictScan. The idea is, when we measure gene expression, there's some component of that is entirely genetically determined. Most of it is attributable to other kinds of exposures and to the feedback of traits and diseases on what we measure as transcript levels. But we can use what we measure in something like GTEC to develop SNP-based predictors of gene expression that then we can test with phenotype. So this is already published last year in Nature Genetics, and all of the software, it's on your bingo sheet, is actually data from GTEC for building the predictors. So we use GTEC as a reference panel to build these prediction equations, and just create a really big database for each gene in each tissue, all of the SNP predictors that come into the prediction equation and the associated weights. We're using ElasticNet for building the predictions. But that means, so once that's created, in any data set where we just have the genome variation, and that can be GWAS level, it can be whole genome sequencing, we can essentially impute transcript levels for each gene in each tissue and associate that calculated endophenotype with our traits of interest, in this case, the medical phenome that we have in BioView. And it has, in any given tissue, there are only about 4,000 to 9,000 genes that have really high-quality prediction. But across all of the tissues that we measure in GTEC, we're getting now high-quality prediction in about 18,000 genes. So and this is with just the first half of the GTEx data that will come in, so that will improve over time. We've also not used ENCODE annotation in this first pass-through in building the predictions. That's something that I think people are looking at and working on. So the quality of the prediction may improve as we can include more information from ENCODE annotations in building the predictors. Plus, we've only built 6 predictors of gene expression at this point. As GTEx matures and we have larger samples, we expect to be able to use at least the top performing trans biology in the prediction equations. So this is all being applied now in the context of the biobank at Vanderbilt University. So Vanderbilt built their own electronic health record more than 20 years ago, and there's a clinical data warehouse, as it were, that we call the synthetic derivative. That's a de-identified, continuously updated, enriched image of the EMR. There's actually a lot of correction that comes in, so outlier variables get looked at hard and corrected. That's about 2.5 million subjects today. We have DNA on more than 215,000 subjects, with GWAS level genotyping on about 20,000 and exome chip data on about 36,000 today. But we're in the midst of some bigger projects. They'll get us, by this time next year, to dense genotypes at about 120,000, and the whole genome or exome sequencing on thousands to tens of thousands, and that's going to be decided by some of the lots of grants that are in the system now. And we'll have DNA on more than 225,000. The synthetic derivative will have more than 3 million observations. These are really rich data. So just the synthetic derivative alone, for learning about the phenome, is a fantastic resource. But being able to look at DNA samples with genome interrogation on tens of thousands to more than 100,000 is a great value. But the really cool thing is the phenome. I had no idea before coming to Vanderbilt how much more science, how much more biology we can learn about disease by being able to look at the entire medical phenome as opposed to individual diseases. And I think Josh Denny, who conceived the concept of the phenome-wide association study, people have thought of it as kind of a curiosity, a cool little twist, flipping a GWAS on its head. It's way more than that, and I'm going to try to convince you by the end of the day that we can learn much more disease biology, considering the phenome in its entirety, than looking at one disease at a time. So the phenome-wide association study, original concept, was taking a single SNP that either, say, the top GWAS finding for Alzheimer's, and looking at all of the phenome associations with that one SNP, or taking a SNP that's a loss-of-function variant in a particular gene, and looking at the entire phenome associated with that. When I say the entire phenome, Josh Denny, who's one of the 41 members of the Department of Biomedical Informatics at Vanderbilt, it's a fantastic resource for doing phenome studies, has set this up so that the tens of thousands of diagnostic codes get boiled down into about 1,600 phenome codes that we look at, and those have been set up to be much more, there's much more information in those than in medical diagnoses, so it takes multiple visits to instantiate diagnosis. For example, it's not, so we also have a number of algorithms for getting to 95% sensitivity and specificity for any given single diagnosis, so this is not that really fine-grained look at a single diagnosis, but it's a much more nuanced look at these 1,500 codes that boil down from the tens of thousands. When we do CWAS, there's a hierarchical organization. So, for example, if you're looking at the phenotype end-stage renal disease, in the controls you don't have kidney failure not otherwise specified, kidney transplant, kidney dialysis. So all of the related phenotypes are not used at all in the analysis. Your control set is a group of phenotypes not on the sort of hierarchical diagnostic spectrum of the phenotype that you're looking at. So keep in mind about 1,600 diagnostic codes. Now, of those, we're not looking at anything that doesn't have at least 30 individuals, and I actually don't look at anything myself that doesn't have at least 50 individuals. So that actually boils down to closer to 1,300. And then a subset of those are normal pregnancy, normal delivery, well child care, sort of part and parcel work physicals. They're codes for billing, they're diagnostic entities, but they're not disease. And so it's less than 1,300 codes, actually, that we're reporting out on. And what we're doing is building this gene by medical phenome catalog. So we're doing not CWAS on a single SNP, but on these predicted gene expression phenotypes, and trying to create a comprehensive gene by medical phenome catalog. You can think of it as essentially knocking down each gene in each tissue and reading out the consequences of that across the entire medical phenome, upregulating each gene in each tissue and reading out the consequences of that across the entire medical phenome. And of course, we're not manipulating the human beings to do that. We're using natural variation, and how we've used GTEx to learn how to read the natural variation and translate that into these computed, genetically predicted transcript levels. I think this makes Bio a really cool discovery engine. And it actually works on the larger scale. So the deviance of the transcriptome is and then genetic density. The only tail of the distribution you ever want to be in is this one, which is the tally of the number of transcript levels where people are at least 3 standard deviations from the mean, plus or minus. Okay? So when you're low here, that's a good thing, because this deviance of the transcriptome is significantly correlated with the burden of medical disease, the number of genome codes people accumulate in their lifetime. And you can see, while most people in BioView have relatively small numbers of genome codes, 15 and 20, there are plenty of people with very large numbers. So when you have a chronic disease, you just accumulate genome codes like crazy because disease begets disease. And there are plenty of people with, yes, very large numbers. I talked before about some of the early findings. So in just 5,000 subjects in BioView, we saw that the reduced genetically predicted expression of this gene, PRIK5, was associated with many different eye phenotypes. None of these are genome-wide significant, so if we do a bond-chronic correction for the number of genes we look at with quality prediction and the number of genome codes and the number of tissues where we have quality prediction, none of these would meet genome-wide criteria for significance. But it's a really interesting pattern, and if a phenotype were just eye disease, yeah, it would meet genome-wide criteria for significance. In such time that I could get the analyses done on the next 15,000 people, they've knocked it out in zebrafish with notable eye phenotypes, and have been able to show in subsequent studies of zebrafish and embryo eyes that the protein product of this gene is very highly expressed in all of the parts of the eye that give rise to the particular eye phenotypes that we looked at. So it's highly expressed in the lens. It's highly expressed in parts of the eye where retinal detachment is going to be an issue. It's highly expressed in the cells that form the sheath, the myelin sheath around the optic nerve, and in parts of the eye that control fluid dynamics. So the association of the reduced expression, genetically predicted expression of PRIK5 with the different eye diseases makes sense given where the protein product is highly expressed. And they've now been following some of these into adulthood and seeing little zebrafish with cataracts in this. So it's a really cool biological validation. But to my mind, the ultimate biological validation of what we're discovering is not with model system knockdown and knockout experiments. It's with the human knockdown and knockout experiments. Mendelian diseases give us a window into human phenotypes that are observed with large-scale knockout of a particular gene across all tissues. And we definitely are picking up with the reduced genetically predicted expression of Mendelian disease genes, a phenotype that you see associated with the Mendelian disease. So there is this continuum from loss-of-function mutations to deleterious amino acid substitutions to just reduced expression of these same genes. For example, here's one nuclear factor 1 X type. Actually, autosomal dominant mutations have this gene associated with 2 different Mendelian diseases, Marshall-Smith syndrome and Soto syndrome 2. They have quite similar features, but some differences. So accelerated bone formation, which then you see as fractures in kids, diminished muscle tone, especially in the upper body, that lead to breathing difficulties. The larynx and trachea are characterized as floppy. They have characteristics.
Some similarity over sequences of different lengths. And so this is what we're going to focus on for most of this morning's lecture. And before we talk about the methods, a little bit about these things called scoring matrices. So once we have these 2 sequences aligned, how do we actually measure how good the alignment actually is between sequence A and sequence B? Okay, so these scoring matrices come into play. They give us a metric to assess how good these alignments are. In the numbers that are found in these matrices, what they actually represent are actual biological characteristics and physical characteristics, or physicochemical properties of nucleotides and amino acids. So these are dependent on the side chain structure and chemistry of the amino acids, their size of function as well. And it takes into account things like, when you look at cysteines and prolines, those are obviously important for structure and function, and especially with prolines, when you find them at the end of an alpha helix, they more normally end up in breaking the chain. Things like tryptophan, you can only put them in so many places because they have a multi-side chain, so as far as steric considerations go. Lichens and arginines, they both have a positive charge, overrepresented in proteins that bind to DNA, so they can bind the negatively charged backbone. But again, taking account charge, shape, all of those kinds of things. So in putting these matrices together, there's 2 major things we're going to think about. Conservation. What residues can substitute for another residue and not adversely affect the function of the protein? Okay. So we know, just empirically, things like isoleucine and valine, they're both small, they're both hydrophilic, they can often substitute for one another. Same thing for serines and threonines, they're both polar. So the bottom line here, the game is to just make sure that you're conserving charge, size, microphobicity, and other physicochemical factors. The other thing that comes into play is frequency. When we look at the entire constellation of proteins and the amino acid composition of those proteins, how often do we see a particular amino acid occur across all of these proteins? So taking into account rare versus common. now, okay, so you now say, all right, Andy, why should I care about any of this? Anytime you do any sort of sequence comparison, when you do a blast search, when you do a multiple sequence alignment, any kind of comparison like this, in the background, these scoring matrices are being used. And the choice of scoring matrix becomes very, very important because they implicitly represent particular evolutionary patterns. So depending on which one you choose or not choose because you decided to use, the default values can strongly influence the outcomes of your analysis. So the default values may not always be the best choice. So that's what we're talking about, how these are constructed, and I'm going to give you some guidelines as we go through. So let's take an easy case first. Let's just look at a nucleotide scoring matrix, and here a simple match mismatch scheme is employed. So if you look at this matrix, the four nucleotides are across the top and down the side. You'll notice on the diagonal, okay, C plus 2. So let's say you had an alignment and you have two C's aligned with each other. Here's the C, here's the C, you see where they intersect. And for that particular position in the alignment, you're going to assess a positive score of 2 points. Every place else off the diagonal, you see a whole bunch of -3. So anytime there is a mismatch, you deduct 3 points. This assumes that each nucleotide occurs 25% of the time, which is actually not true, but it's close enough, and that's what's usually used. And this kind of scheme actually works okay because, in general, the nucleotides are not all that chemically dissimilar. But when you compare them to amino acids, the situational landscape becomes much more complex. So this is an example of a protein scoring matrix. It's something called BLOSUM 62. This is the default matrix that is used for the blast searches at SGBI. You'll notice across the top, we have all of the amino acids across the top and going down the side. So let's take a couple of cases so you can see how this is different from what happens in the nucleotide world. Let's say again you have sequence A and sequence B, you've aligned them, and there's a particular position where you have two tryptophanes lining up with each other. So we're going to look for the W up here, the W over here, and see where they intersect. And in this case, for that particular position, because it matched, the value that we assess is 11 points. Now what I want you to do, and you can probably see this better on the screen, look at the diagonal. And you'll see that unlike the nucleotide case where a match was always assessed the same value, here it's not. Okay, so here we're taking again into account how frequently these amino acids occur in the entire constellation of proteins.